Proteins are actually chains of units called amino acids. Amino acids are mostly made up of carbon, hydrogen, oxygen, nitrogen, and a couple of them will have sulfur. Their function, main one, is that they will act as organic catalysts. A catalyst in chemistry is a substance that will increase the rate of a reaction. In chemistry, a lot of times these are dangerous because they can increase the rate of reaction so much that it produces lots of energy all at once, such as heat, or sometimes even an explosion. Now, our enzymes act as catalysts, but they can be regulated, and therefore we can maintain homeostasis. Without these, the enzymes we use to digest our meals, like after lunch, some studies have shown where you might have to heat the stomach up to 350, and some say maybe, depending on what you eat, 700 degrees in order to chemically process it without enzymes. And of course that would be deleterious to living tissue. So it speeds up the rate of the reaction, but again, while being regulated. A lot of these also add structure, such as girders and frameworks inside of cells and even outside of cells. And also they can be burned for energy. Just like, again, think about structures. If you're in a winter cabin, you're running out of firewood, you can break up the furniture. So you can burn these for fuel, and a small percentage of your energy every day actually comes from burning some proteins. At times you can increase the amount of proteins you burn. A lot of times you don't want to, because your major source of protein is muscle. And that's why just dieting, you're going to burn more fats, and you're also going to store more fats. And so you'll lose weight, but what you're actually losing is usually muscle. And you're putting on a little bit more fat, but you look a little bit thinner. The structure of a basic amino acid, here's a better example, better than my drawing. Central carbon, and off of that we call this an amino acid because off the side here is an amine group. That's like ammonia NH3. This is NH2, and the third bond rather than a hydrogen connects to that central carbon. Amino acid over here on the side of this carboxyl group is actually an acid group. Now, this oxygen next to a hydrogen, remember, polar covalent. The oxygen is strong enough to pull that electron more into the oxygen, but not enough to take it away totally from that hydrogen. However, with the help of this second oxygen through this carbon, collectively the two of them can at times act together and pull that electron off here, releasing that hydrogen, of course, now as an H+, making it an acid. Now, it only does that about 30% of the time, and so it makes it, remember, a weak acid. In the center here, the carbon needs 4 bonds, so the one here is a hydrogen. And also down here, though, the fourth one is what we call the R group. Now, as you probably know, there are 20 different amino acids we use to link together in chains, short chains or longer chains, in order to make proteins. The 20 different amino acids have this all in common. What differs between the different 20 is the R group. This is what varies and was referred to as the variable group. Okay, so R stands for variable. And if it doesn't make sense, that's okay. Back in the day, the R group stood for remainder of the molecule or rest of the molecule, which kind of makes sense. Sometimes it's just a little CH3 group down here, and one it's a hydrogen. Another one it's a large organic portion, lots of carbons and hydrogens, okay, maybe a nitrogen. Sometimes these R groups are large, sometimes small. Sometimes hydrophilic, sometimes hydrophilic. Depending on the pH, some will be positively charged, some negatively charged, and many of them will be neutral. So they actually add a lot of the chemistry to the amino acid. We can join the amino acids by simple water chemistry. Here are 2 different amino acids, and you'll remove H off of one, OH off the other one, in other words, water. And this nitrogen will now combine with this carbon. If we put this into a little bracelet of amino acids that's called a peptide. A long necklace of these is a protein. Some books will say less than or greater than 50. Some will say less than or greater than 100. What I was taught was 100, so that's what I go by. Some of these long proteins can be 2,000 amino acids long. That's a pretty long necklace. Of course, first day of class, we even talked about structure with function. And this is one of the best examples of structure with function. Now we have different levels of structure because these will fold up into three-dimensional structures, and of course that structure gives their function. The first level, what we call the primary level, it means the same thing, is simply the order the amino acids are put together. Which of the 20 is number 1? Which of the 20 is number 2 and 3? And of course, if we're getting above 100 or even close to 100, you're going to have repeats in here of at least some of them. And so that's the order of these. Now that's our simplest structure. Now remember, with some of those NHs and OHs and that, we're going to have actually hydrogen bonds form. And so when you have those partial positives and partial negatives along that amino acid chain, they're going to interact, and a lot of times they form two basic structures. They can form alpha helixes, and so there's an alpha helix, a little twisty, kind of like that old-fashioned cord that led from your handset to the phone. Okay, kind of like that. And the other one's called a beta sheet. The cord you plugged from your phone into the wall, zigzag like this when you unfold it. That's a beta sheet. These create what we call domains, or parts of the protein. That's a secondary structure. Tertiary structure, third level structure, is the interaction of those R groups. Some are hydrophilic, so they're going to hang out here in water. Some are hydrophobic, so they're going to cluster together like oil droplets. Some are positively charged, and other ones are negatively charged, so they're going to come together kind of like a magnet. And ones that are like charges, they're going to repel and push away. And so all this leads to a three-dimensional structure, and of course that structure leads to what's going to be the function of this protein. Some even have a fourth level, a quaternary structure, where proteins actually will interact to function collectively. Best example is one from AP2, but hemoglobin. 4 chains of proteins all linked together. Of course, its job is to transport oxygen. They hold inside them a heme group and an iron that holds oxygen. In your lungs, doesn't matter which of the 4, any one of these 4 binds to oxygen, it changes the shape of the protein, changing the shape of the ones they're directly linked to, and ultimately the one they're indirectly linked with, increasing the affinity of their attraction to oxygen. So not just one of the 4 proteins, but all 4 will pick up oxygen in the lungs, making it a lot more efficient than if they worked alone. So now they're carrying 4 oxygen through the body, and they get to a tissue that needs oxygen. And so it's using up its oxygen, it's low on oxygen, so via diffusion, any one of these oxygen that comes off will change the shape of the one chain, which changes the shape of the other 2, and finally the 4th, and decreases their affinity so it doesn't drop 1 oxygen but 4 oxygen to the tissues it needed, making the delivery of oxygen a lot more efficient than if they worked individually. That structure is so important that if you take it out of its natural state, it will no longer function, at least not very well at first, and then not at all. In fact, there comes a point where it cannot go back to its original shape, and so it's ruined. So it either has to be recycled, and at least it has to be remade. It needs to be replaced. And that's called denaturing it, taking it out of its natural shape. You basically unwind it, you scramble it, you mess it up. A few basic ways you do that. One is heat. We already talked about that. Remember that little scenario? This is your brain on drugs. Remember that egg, especially that egg white, starts out as clear, transparent fluid, and very good for that baby chick to use as a food supply. And once you add heat is kinetic energy. Those molecules of albumin start moving faster and faster and start unwinding, start spinning, and start interlacing with other ones until you get a white, opaque, solid mass, basically a scrambled egg. And that is not a good food supply for the baby chick anymore. It is for us, it's better for us, but we can eat it as a raw egg or as a cooked egg, but it doesn't have that function for that baby chick anymore. Keep in mind, not the best example, but remember we talked about that body temperature of 108 for a sustained amount of time. Your blood proteins are starting to be affected and will make you sick. pH. We said those R groups and the way they're attracted, whether positively charged, negatively charged, or neutral, is actually based on the pH. And usually within a certain homeostasis, such as in the blood, remember 7.35 to 7.45. You go outside that, some of those R groups are going to change from positive to neutral, okay, negative to neutral, maybe even from negative to positive. And if that happens, they're going to start now not having the same structure and start to change. They will not function well, or maybe not at all. And again, if it's permanent damage, it needs to be replaced. Next one is radiation. And of course here we're talking about gamma radiation. And gamma radiation, remember, it can cause chemical reactions that normally don't occur. So when you zap a protein, it can cause a round protein to actually flip around on itself and have a totally different shape if it hits it just right. Actually, in radiation poisoning, radiation sickness, there's two levels of it. One of them is immediate, and that's what you get from protein damage. This is what causes welts on your skin, internal bleeding, is when these proteins now get damaged so much that they can't do their jobs. Now in this case, what you're going to have is actually you need to replace it. And so the race is on whether you're going to survive this radiation poisoning is if you can get back to homeostasis. You know, if you don't get very much radiation, you don't do much damage, okay, not a problem. But once you exceed a certain amount of damage, now you need to repair that damage, replace those proteins, and the clock is on. Now the secondary damage from that radiation is it also can damage your DNA, and if you damage the blueprint part of this that is going to cause the repair, then you have a bigger problem. That's your long-term damage. And damage too many things or key things and you can't repair it, and so basically you're sick at first, and then if you can't repair it, you die. Chemicals. Urea. Urea. Now, when you burn amino acids, we've talked about, well, we will talk about again, but in AP1 you learned, you know what to do with the carbon, the oxygen, the hydrogen, but you don't know what to do with that nitrogen. Now, fish, amphibians, lower life forms, what they do is they pluck off that amine group and just releases ammonia. And most of them are aquatic, and so it just dissolves into the lake or the ocean or the pond or whatever, and nature takes care of it. We don't do that. Ammonia is very toxic, and so we take that amine group, actually two of them, and we put it on carbon dioxide, and that makes urea. It's less toxic. Now, it's still toxic to a certain degree, but once it gets put in the bloodstream, filtered out through the kidneys, mixed with water, it's urine. If you do not filter it out in a certain amount of time, your kidneys shut down. You need dialysis to remove it, because if you don't, within a couple of days, it will reach toxic levels still and kill you. Again, just less toxic.